Hello, I'm Moses Oreto. I'm a teacher of biology, and I would like us to discuss something in Form 4 class, and that is genetics, the first topic in the Form 4 class, and that is genetics. I know that we were introduced to genetics while we were in Form 1, and we learned a lot in genetics. And of course, from the definition, we were told that genetics is the study of inheritance and variation, which is all about hereditary materials that are supposed to be passed from the parents to the offspring. In this concept of genetics, I want to be more specific, and I want to discuss a concept on monohybrid inheritance. In monohybrid inheritance, in this lesson, some of the objectives that we need to achieve by the end of the lesson, the first objective, we have to discuss something to do with the monohybrid inheritance. Monohybrid inheritance. And the lesson objectives of this lesson. 1, by the end of this session, the learners should be able to define a monohybrid inheritance. A learner should be able to define monohybrid inheritance. That is the first objective that we need to achieve. The second objective, the learner should be able to differentiate between genotype and phenotype. The learner should be able to differentiate between genotype and phenotype. That is the second objective. The last objective that a learner should be able to capture is to draw a diagram to show genetic crosses. The learner should be able to draw a diagram to show the genetic crosses. The diagram to show genetic crosses. So those are the objectives of today's sessions that we have to capture. I want to start by defining what is monohybrid inheritance. And I want to say about the monohybrid inheritance, the monohybrid is about the passage of a single characteristic controlled by a pair of genes. We are being told that gene, or what we call the genetic materials, is what is defining the characteristics of every living organisms. And this concept, because these are the genetic materials that are supposed to be passed from the parents to the offspring, and these genetic materials, they are contained in a specific locus within a chromosome. But for they to be passed from a parent to the offspring, of course, we know that there's that one that is supposed to be from the male side, and that one that is supposed to be from the female side. And they must be passed in pairs. And that is why I'm referring to monohybrid as the passage of a single characteristic controlled by a pair of genes. So it therefore means for this characteristic to be passed, it must be passed in a pair or in a pair of genes. And therefore, in the definition is what is going to guide us, so that when we will be doing the crosses, we'll be drawing the crosses to show how these genes are supposed to be passed, then from the definition will be guiding us so that we'll be able to capture it or have it in the right way. Then in the next thing, this monohybrid inheritance, because we are going to look it in the concept of how we can present it in genetic crosses. But before we do that, because there are those basic things that we were introduced to when we talk about inheritance, there are those specific terms that we really need to know, or we need as a learner, we should be able to know, so that when we will be doing the genetic crosses, you should be able to be guided accordingly so that we don't lose the concept. One of the things that we need to know, because these things is all about the gene, there's this concept that you are told about the genes. The genes can be of two categories. We have those that are being referred to as recessive genes. We have those that are being referred to as dominant gene. Therefore, we should be able, or the learner should be able to differentiate between a recessive gene and a dominant gene. What's this thing that I'm calling a recessive gene? If I'm talking about a recessive gene, then I'm referring to a gene that is suppressed by another gene. Remember, these genes occur in pairs. And if you have to present them in pairs, for instance, I want just give a good example. We might be having a gene, because if you have a chromosome, we have the chromosomes from the female side and the chromosomes from the male side. And each must donate one. And therefore, after donating ones, they are going to be in pairs. So for instance, I want to talk about the gene that is controlling maybe the height. The gene that is controlling the height, we are being told that if you want to present the genes, you use the letters. And the letters that we are going to use, you can use any letter to represent them unless you've been told or you've been guided to use a specific letter. For instance, I want to use a specific letter to represent the gene for height. I want to use capital T, small t, small t. This is the uppercase, this is the lowercase. The capital T is a gene that controls the height, as well as the small t is another gene that is controlling height. But what is the difference? The difference only comes in whereby one is a dominant. The one that is represented by capital letter, we call it dominant gene. We call it dominant gene. And the one that is represented in small letter, we call it a recessive gene. So, when they occur, for instance, for this case, we have pure dominant gene, where we have capital T and capital T. So for this case, we say the gene here represented is called homozygous tall. This one represents the tallness. This one represents the tallness, while this one represents controlled. Let me use the word control. This one controls the shortness. So for this case, we have these genes, the one that is controlling the height. One controls the height for the tallness. Another one controls the height for the shortness. So for this case, if you have the two T without the mixture, they are purely uppercase, then we say this is homozygous dominant. This is homozygous dominant. So homozygous dominant, you should be able to get that. And the other side, where we have another pair of gene that is controlling the shortness, this one we refer to as homozygous recessive. Homozygous recessive. While this one is homozygous dominant. So what I was trying to bring into your attention, that we must be knowing about two types of the gene that can be represented under genetics. The gene can either be dominant or the gene can be recessive. And I've said A dominant gene, if I have to repeat, a dominant gene is that gene that is suppressed, the other gene. And the other gene here is a recessive gene. While a recessive gene is a type of gene that is suppressed by a dominant gene. But look at here. Here I've represented a pure dominant gene, while here I've represented a pure recessive gene. But sometimes you might find that the two genes can be mixed, or during the process of passing of genetic materials, the two genes can get together, whereby instead of having pure dominant and pure recessive, we have a dominant gene and a recessive gene. Now, how do we call this category of gene? For this case whereby we have a mixture of both dominant and recessive, this one we refer to as heterozygous. This one we call it heterozygous dominant. Heterozygous dominant. So this type of existed whereby we have got a mixture of both dominant and a recessive gene. The biological term that is used to refer to this concept is referred to as heterozygous dominant. So we should be able, and for this case, you will be told, where we in a case whereby we are having both the dominant and the recessive genes coming together, what is going to control the physical appearance of that particular organism is the gene for the dominant. It is going to suppress the recessive gene. So I want to say, this one we'll come to look at it later when we'll be doing the genetic process, so that we'll be able to understand how is this one taking place. But then we should also be able, now, because when we'll be doing the genetic process and the so on, and maybe other things that is related to it, we'll be coming other terms. We'll be coming across other terms. We'll be coming across other terms, and some of the terms that a learner will be able to come across. Some of the terms that a learner will be able to come across while doing this kind of concept. One of the things that a learner will be able to come across is called, we have the term phenotype. And we have also the term genotype. So these are the two terms that we are going to come across while we are going to deal with this concept of monohybrid inheritance. So a learner should be able to know what is this biological term referred to as the phenotype, and what is this thing that is called the genotype. So if I have to differentiate the two terms, or to make learner know the difference between the phenotype and the genotype, a phenotype is the physical or observable feature or the outer appearance of an organism. What you'll be able to observe from the outside, the observable characteristics in a given organism. So for instance, I want to give a good example. The skin color of a human being is physical observable characteristic in a given organism. For instance, it could be human being. Somebody could see a particular human being and say the skin color is black. Another person can also having a brown skin color. So what we will be able to observe, the physical or the physical appearance of that organism, that is what we are going to call it, or what we are going to refer to a phenotype. That is the physical observable characteristic of a given organism. It could be in terms of flowers. Some flowers are pink, some flowers are yellow, others are white. That is a physical characteristic that we are able to see from outside, and that is what we call the phenotype. Then again, there's a concept called genotype. What is this thing called the genotype? If I talk about the genotype refer to the genetic the constitution, or what constitute the genetic makeup of a given organism. The constitution of the genetic makeup of a given organism is what we call the genotype. Or just in a simple term, you can refer to it as the genetic makeup. Genetic makeup of living organism. Genetic makeup of living organism. This genetic makeup of living organism is what is controlling the characters, or what is going to help us know the physical appearance, or they are going to express certain physical appearance that somebody can be able to see, and maybe from there we'll be able to know the phenotype of that particular organism. So from now we should be able to know the difference between the phenotype and the genotype. And this case of genotype, which is the genetic makeup of living organisms, this genotype, it is always presented in pairs. It is always presented in pairs. It is the one that is controlling the characteristics of living organism. Must be presented in pairs. And that one we are going to see when we will be doing the genetic crosses and the rest. But in this this concept of monohybrid, before we do the genetic crosses, there's also a very important thing that we should be able to know. There are those things that must be there. Those things that must be there in carrying out a genetic cross. Those details must be known to the learner, or the learner should be able to know them so that you do the genetic cross in a better way. So what are some of the details that you need to learn, or you need to know, so that when you'll be doing the genetic crosses, you'll be doing it in the correct way? First, we have to know the symbol, because here involves the male and the female. We are always using the symbol to represent the male and the symbol to represent the female. So details required in genetic crosses. Details required in genetic crosses. What do you need to know? One of the things that we should be able to have is the symbol. The symbol to represent the male and the female. Representing the male for the male character and the female. So what is the symbol that is used to represent the male? We always represent the male under genetic crosses by having something like this, but ensure that this line is well drawn, the circle is well drawn. You have to use your free hand, and the other line that is extending, that is showing the arrow, must not pass the circle, getting inside the circle. It should only just touch the edge, and it should not be left hanging. It must touch the edge of the circle. This one represents the female. And if you want to have the symbol for the female, this one represents the male, sorry. And if you want if you want to know the one that is representing the female, you'll have a symbol like this one. That one representing the female. So those are the things that you all need to have when we're doing the genetic crosses. Apart from that, we also have to have something to do with the phenotype. We have to have something to do with the phenotype. We must ensure that the phenotype is included. The phenotype is included. We must ensure that the phenotype here is what you'll be able to see from outside. Maybe from outside, this particular organism maybe is dark in color in terms of skin tone, or in terms of height. These organisms, you'll be able to say this organism is short, and you have to indicate that one in writing. So that is the observable outer appearance, or what we'll be able to see from outside. Then after that, we'll be also have something to do with the genotype. we must include the genotype. And the genotype here is about the genetic makeup. And I've said before, the genetic makeup of living organism must be presented in pairs. The one for the male and the one for the female. We are going to see how they are supposed to be presented in pairs. Then after that, we must also include something to do with gametes. And the gametes, we are getting the gametes after segregation of the genotypes. After the separation of the genotypes of the male and that one of the female. That is what is going to give us the gametes. And these gametes, they must be circled. You have to circle the gametes. After this separation, they must be circled. Then after that, we also have to know or to represent the process of the fusion. And the fusion here is all about how the male and the female gametes are combining to give us now the F1 generation or the first foliar generation. And that is what last we are going to have. First foliar generation. This is the last thing that we are going to represent in this concept that you need to have. So from here, I want to give out a good example so that we'll be able to know what I'm trying to do. For that learner who is listening at home, I want to give this good example so that we'll be able to calculate it, so that we'll be able to see all those concepts that I'm talking about when I talk about the monohybrid inheritance. We see how a pair of gene is being passed from the parent to the offspring. Then, in this case, maybe I want to read a very good example. There's an example here to show the monohybrid inheritance. I will just read. In a garden pea, the gene for red flower is dominant over the gene for yellow flowers. The gene for the red flowers is dominant over the gene for yellow flowers. Pear plants with red flowers were crossed with the pea plants having yellow flowers. Both parents are homozygous. Both parents are homozygous. Work out the genotypes of the offspring. Work out the genotypes of the offsprings, hence represent the F1 generation. So from here, if you are listening keenly, we've been told that we have a garden pea. The gene, one is red, having red flowers. Another one is having yellow flowers. So we have two types of plants, but with different flowers. That is now the concept of genetic crosses. How we can represent it in a diagram, genetic crosses. How you can represent it in a diagram. Of course, I will actually maybe tell you that we have got two ways of representing it. Somebody can use the genetic crosses. Another one can use what is called a Punnett square or a Punnett square. So for this one, we just want to learn how we can represent the genetic crosses. Maybe later on, you will be able to also know how the same can be represented in a Punnett square. So I've said that we have a garden pea. One is with red flowers, another one is with white flowers. Then, for instance, we are being told the red flowers is dominant. And I've told you about the dominant gene. If I talk about a dominant gene, it must be presented in uppercase. And that is in capital letter, depending on the letter that you've chosen or the one that has been specified for you. So the dominant gene is red flowers. which is dominant over the white, over the yellow one. Sorry for that, over the yellow one. So we have got two flowers, the red flower, and we have the white flower. The yellow one, sorry, I am referring to white. The yellow flowers. The red flowers, we've been told, they are dominant. They have dominant genes. While this one is having recessive genes. Why am I saying this one is having recessive genes? Because from the concept that you've been told, from the question that I've just presented, we are being told that the red flowers is dominant over the gene for yellow flower. Then if this is dominant over that, what does that one means? It means that this one must be having a recessive gene. And for that case, our problem is going to be solved. Already it has been stated for us. This one is dominant over this gene. And therefore, the gene for the red flower must be presented in capital letter, while this one is going to be presented in small letter, but for the same character, for the same characteristics, for the same characteristic, which is controlling the color of those flowers. So let us check at this one. At the same time, you are being told there's a cross was made between those flowers having yellow flowers, with the plant having yellow flowers, with the plant having a red flower. But both parents are homozygous. Both parents are homozygous. If you've been told that both parents are homozygous, meaning they have the same gene for that particular characteristics. We don't have a mixture of genes. And therefore, I want to give any letter to represent the dominant gene. Then the other letter, which is on the lower case will represent the recessive gene. Look at it. Let us, the gene that is controlling the flower color, because here we've not been given. Let the gene that is controlling the flower color, let's pick R. So we are going to have capital R, and we are going to have small r. So this capital R is controlling, or I could have used a different letter because somebody will think R will be red, while the other R will represent a different color. Let me come up with a different letter. Let me say the letter can be C. So capital C to represent a dominant gene, while this one represents dominant gene, while small c to represent recessive. Remember, the two genes are controlling the same character, which is the flower color. Only that one is dominant over the other. So let's pick it from here. How do we go about it? Now we've known the one that is dominant and the one that is recessive. So let us now go directly to the genetic crosses. Let's now go directly to the genetic crosses. So I will start by this. Remember, these genes are homozygous. Both parents are homozygous. And therefore, this one will have pure dominant genes, which is capital C. Because we've already been told that all of them, they are homozygous dominant. While this one will be having small letter c, because that is the one that is controlling the recessive gene. I said that they must be presented in pairs. Now we come to this point. Now we want to represent them using the genetic crosses. We start with the parental or the parent phenotypes. Parent phenotype. The parent phenotype here, of course, we'll have to know the symbols to represent the male and the symbol to represent the female. So I'll be having my symbol just slightly above the parent phenotype, just slightly above it. So I'll be starting the one that is representing the male. Then this one representing the female, this side. So I want just say, let the one with the red flowers to be the male, or you can say it is the female. It doesn't matter unless you've been it is the one that is having the red flowers. So from the outer appearance, I will say it has red flowers. So I'll just mention what I'm able to see. So I just say it is having red flowers. I just write red flowers, or the red flower, because I'm using one of the male. Then the other one, I represent it with yellow flower, because I'm just using one female to represent the rest. Then after representing, remember, we have to put the colon before those ones. Ensure that you don't forget to put that one. After that, I go to the parent genotype. The parent genotypes, this is the parent phenotypes. Now we go to the parent genotypes. Remember, you have to put the colon. Now the parent genotypes are now the gene, which is the genetic makeup. The genetic makeup of that particular organism. The genetic makeup of the red flowers, those with the red flowers, is capital C, must be presented in pairs. The one that is the genetic makeup with those with yellow flowers are represented in small letters. So we are going to have capital C, while the other one is going to have small c, small c. But what we should not forget, where are we supposed to be representing the cross? Because it's about the genetic crosses. So we must have this, and that is how we represent our cross. Where are we supposed to be placing our cross? The cross is going to be placed where we have the parent phenotypes. And this is where we are going to have our cross at that particular point. So let us not forget, because some student might forget, they place it on the upper side. Some student might forget placing it on the lower side. So this is the appropriate place for having the genetic cross. After this, what else are we going to have? We are going to have the gametes. Or we can have the parent gametes still. Ensure that we have that one. So how do we go about it? The gametes, just the same way I said, we must have the segregation, the separation of the male genotype and the female genotype. So during that, we must have something like this. And please check on how I'm doing it. Check on how I'm doing it. So we are going to have capital C at this point, but we circle it. As you circle it, ensure that this line that is showing the separation of these genotypes, it must touch the circle where we have our gamete. Then another capital C at this point, and we circle it. This line just should touch capital C, and we circle it. It should touch the edge of the circle. If that one doesn't happen, then it will be considered as a wrong diagram. So those ones must be observed. Then the same thing will be done to the other side. The other side, small c, we circle it. Small c, we circle it. So up to that point, we have represented our gametes. All of them is circled. So ensure that that one is well done. Thereafter, we check on something to do with the fusion. We check on something to do with the the fusion. And the fusion here just simply means the union of the female and the male gametes. The union between the female and the male gametes. So, for instance, we have the first one here. This first one, which is the first gamete here, can combine with the other gamete, the first gamete on the female side. So if the two combines, what are we likely to get? So we are going to have the first one combining with the first male gamete, combining with the other female gamete. And in that case, what do we have here? According to the order, it is always the dominant gene coming first, followed by the recessive gene. And therefore, if I have to represent it, I'll be having capital C first as the dominant gene, followed by the small c, which is a recessive gene. So this is a possibility that when the two combines, then there's a probability we'll be having a flower with this particular character, or with these particular characteristics, I mean. That is the first case. What about the second case? The same gamete, the same gamete, that is the same male gamete that combined with the first one, can also combine with the other one. If it combines with the other one, what do we get? We are going to get capital C again and small c. So that is another possibility. Remember, we are not carried the one for the other gamete. The one for the other gamete after separation, we are only done with the first one. There's also another chance that for the other one also, it can combine with the first one. And if it combines with the first one, we are going to have capital C, small c. Capital C, small c. The other one as well can also combine with the last one, also to have capital C, small c. So these are the chances of what we are likely to get as a result of the union between the male and the female. When the female gametes and the male gametes come together, these are the probability, the chances of what we are likely to get. So what we are going to get here, these are what we call F1 generation. F1 generation, which is the first foliar generation. Those are the offsprings that are going to result as a result of the first mating between the male and the female. So this is what we are going to get. And this is, after getting this, remember from our question, we are being told, work out the genotypes of the offspring. And if you have to work out the genotypes of the offspring, it's like somebody telling you to get the first foliar generation. And if you have to get the first foliar generation, this is what we are likely to have. This is what we are likely to have as our first foliar generation. But what do you see from the end of it? The probability or the chances from the union between the red flower and the yellow flower, the chances are all of them are going to have the same genotypes. All of them are going to have the same genotype. And somebody might ask you, from the presentation or from the genetic cross, you may be able to conclude on certain areas. Of course, I've talked about the genotypes, and I've said all of them are having the genotypes of C, small c. Remember, the genotypes must be represented in pairs. So all of them will be having the same genotypes. This is now the genotype of what we have. And what about if somebody asks you about the phenotype? If somebody asks you about the phenotype is the observable feature of this organism. And the phenotype here, if I have to talk about it, what we'll be able to see is that when I have the capital C, small c, remember capital C is a dominant gene, while a small c is a recessive gene. And therefore, what we are going to say? When a situation whereby we are having a dominant and a recessive, just the same case, if I have capital T, small t, this one is a dominant while this one is a recessive. Remember, this one will suppress the other one. And therefore, we are going to conclude that this will suppress the other. And phenotypically, the T will take the day. The capital T will take the day. What the capital T was controlling. So from here, all the F1 generation will be red in color. And that is the case. I've just given the first scenario, and therefore, from whatever I've done, I think we are able to achieve our objectives, the three objectives. That is, we were to define what is monohybrid inheritance. We were to differentiate between phenotype and the genotype. And we were supposed to represent the genetic crosses. And I've given one example of the genetic crosses. Maybe you as a learner, I might maybe give one example you go and try at your own time, so that you'll be able to see whether you'll be able to follow up what we've learned. But this one, we are using the genetic crosses. And let me read out this example. Let me read out this example to the learners. A cow with white fur was crossed with a red bull. No, this is a different one. Let me read the one that is concerning the monohybrid inheritance. A rat, in rats, comma, the allele for black fur is dominant over the allele for the brown fur. Work out the first foliar generation using genetic crosses. Work out the F1 generation using the genetic crosses. So that is another scenario where we have the rat. If I talk about allele, because here they have talked about allele, allele is just the alternative form of the gene. So there is one with a black fur, which is dominant over the one with a brown fur. So we are being told to work out the F1 generation using genetic crosses. So that is another simple scenario. And thank you for listening. We know that if we'll be having another time, we'll be able to work out the others, as well as representing the rest in a Punnett square. So that is all. So this is a very good opportunity for the learners to gain something. Let us not just rest at home. At least we should be able to tune in so that we'll be able to get something by the end of the day. And I know that those who have been listening, I know that there's something that you've gotten and it will be useful, maybe in one way or the other. And let us also, for those who are watching, let us encourage others also to do the same, so that we make a good use of time. We make good use of time. And thank you so much for your time. And that marks the end of our lesson.